In this next section, we're going to look at some examples of epithelial tissues and figure out how to distinguish and identify them. So let's start off with the easiest, which are the simple epithelial tissues. Here we have the amnion, the start of the amnion, what starts and becomes the amniotic sac. Here we have right here, you see one layer of simple squamous cells, nice and flat, very wide. And then what we see below it is a different kind of tissue. So this one layer of cells right here, that's simple squamous epithelial tissue. Over here, we have a row of cells here, and then what's below that row of cells looks different. So it's not part of the same tissue layer. These are kind of squarish, and remember that when tissue gets sectioned to make a microscope slide, it doesn't get sectioned perfectly straight across the middle of a cell or through the middle of a tissue. And so we don't always see exactly the shape we're expecting. But these are all sort of about the same width as height, and so we would categorize these as cuboidal cells. There's one layer of them here, one layer here. The space inside is a lumen. So this is simple cuboidal epithelium making up the walls of a tubule inside the kidney. So when you see a space like this, don't think that that's part of the tissue you're looking at. Okay, so remember, you're going to look first at the number of layers and then at the shapes of the cell. Now, when we get to columnar epithelium, we can have cilia present or absent, and in some cases, we're going to have microvilli. So this tissue sample here, and here, this is the non-ciliated simple columnar epithelial tissue. So this whole thing, that's a single cell. These big things that look empty, those are called goblet cells, and those are specialized cells that release mucus. And they sit in between the much narrower columnar cells. The reason that this edge looks kind of fuzzy is because of the presence of the microvilli. The way you can tell that this is simple columnar is that all of the nuclei are neatly lined up at the basal side. Now, they're not always quite so neatly lined up, but it's definitely one row of nuclei. And then deep to that, something else. Okay, so that's definitely non-ciliated simple columnar epithelium. Over here, we have similar looking tissue, but this time we see really clearly a layer of cilia. Now these cells, you can see they're very tall and narrow. There's one row of nuclei all the way around. Now over here, we get to where we see like maybe a few, maybe kind of smushed together. But in general, what we see is one row of nuclei. Again, this could be because the cells got sectioned in such a way that it looks like there's two nuclei or two cells or two layers of cells. So look at the general, the average of the cells, and you'll see one row of cells. So this is simple columnar epithelium. We can clearly see the cilia, so it's ciliated simple columnar epithelium. And then all of this, of course, is the lumen of, in this case, the uterine tube or the fallopian tube. Now there's one other type of tissue that is going to try to lead you astray, and that is called pseudo-stratified columnar epithelium. It is almost always ciliated. It's pseudo-stratified. Pseudo means false, which means that it looks stratified, but it's not. It's a fake stratified tissue. So there are a couple of different things to look for. One is if it's ciliated, it's always one layer of cells. Okay. If you think about it, if there were another layer of cells here, if this were actually stratified, it would have to be one layer of simple ciliated and one layer of non-ciliated simple columnar tissue. So you can't have two layers of ciliated tissue. One layer would have cilia up against the other one, and that wouldn't make any sense. So here are the cilia, so that means we're looking at one layer of cells. Now, in this diagram, you can see, like for example here, there's one cell that goes all the way up to here, okay? But sometimes it can be really hard to see. So one of the things you look for cilia. If it's ciliated, it's either simple or pseudo-stratified columnar epithelium. If it doesn't look this obviously simple, then just say it's pseudo-stratified, okay? There are very rarely, really, I think the male urethra is the only one, stratified columnar epithelium. Notice it does not have cilia. So if you see cilia, it's definitely not stratified columnar epithelium. It's definitely pseudo-stratified. And here you can see really clearly, you can see the distinctions between the cells. Now, obviously, this is a diagram, okay? So when we look at real tissue, what we see is this. So the first thing you look for is what shape are the cells, so we know whether we're looking at columnar, cuboidal, or squamous. And these are obviously tall, thin cells in all of these. And even here, you can tell that it's kind of hard to tell whether is this all one thing? You got to look pretty carefully, but they all look like they're pretty tall and thin. All right, so we've got columnar here. Okay, so is it one layer or multiple layers? This one looks like it's very clearly one layer. You can see a row of nuclei lined up here. That very clearly looks like a row of simple, non-ciliated columnar epithelium. That's what this is. This, you can clearly see the cilia. And we see down here kind of a row of nuclei, and then almost looks like two rows of nuclei in some of these places. But remember, if it's ciliated, it's not stratified. So this is pseudo-stratified ciliated columnar epithelium. This one is actually stratified columnar epithelium. So you can see down here the type of cells change. This is a row of columnar cells. This is another row of columnar cells. Now, you may be thinking that's pretty subtle, and I don't know that I would have noticed that. What we're working on here is training your brain to learn how to learn what these look like, okay? You're not going to get it right away. You have to look at lots and lots of samples. And in another video, I'm going to show you some strategies for how to do that. But you got to practice looking at things and then break down, what am I looking at? What shape are the cells? Is there a lumen? In which case, it's definitely an epithelium. See, there's a lumen here, lumen here, lumen here. And how many layers of cells are there? Can I tell? Are there other things that help me distinguish, like the presence or absence of cilia? All right, now, squamous tissue is very often stratified. And we have one other thing we need to add, and that is the presence of keratin. Your skin has keratin in it. It's called keratinized stratified squamous epithelial cells. Keratin is a protein. It's very strong. It's the stuff that makes up your hair and your fingernails. And all of these cells in the epidermis of your skin, this upper layer, these are all filled with keratin, and that makes them A, very strong, and B, mostly waterproof. So keratinized stratified squamous epithelial cells look darker on a microscope slide, and they look more solid, okay? When we see non-keratinized stratified squamous epithelium, the cells look more empty, and that's because they're not full of keratin. So this is the wall of the vagina. We see similar tissue in the rectum, in the mouth, and in the esophagus, which is the tube that you swallow food down. We see the same thick layer of stratified squamous epithelium. Now, if you look down here, See, it actually sort of starts to look like cuboidal cells. And in fact, actually, this part is cuboidal. I need to change this because this row is cuboidal. All right, so non-keratinized versus keratinized. Keratinized, solid-looking cells. Non-keratinized, empty-looking cells. Still stratified squamous. Look for the nucleus and then look around at the shape of the cells. You can see they all look really flattened. Now, we do have stratified cuboidal epithelial cells. And in this case, here's the lining of, this is a duct. This is the wall of a duct of some sort. Here's the wall of a sweat gland. And you can see here, we do see 2 layers of nuclei. In some places we see 1, and even here we see 1 as one other cell. It's either sectioned oddly or another cell is being made to replace it. But generally we see 2 layers. Some places looks like maybe there's 3, so it's definitely stratified. And all of these cells are they don't really look flat. They don't really look columnar. If you just looked at this section here, you might think they're columnar. But when you look at a wider area, you see, no, they don't really look that columnar. They look shorter. So this is definitely cuboidal. So 2 layers, stratified, kind of squarish, roundish cells, cuboidal, stratified cuboidal epithelium. Now, one weird kind of tissue is transitional epithelium, and we see this only in the urinary tract, in the ureters that carry urine from the kidney to the bladder and in the bladder. So it is an epithelial tissue. It's up against a lumen. And the cells look like stratified cuboidal mixed with stratified columnar, kind of. And they look a little different than cells in other places. One of the reasons is that these sometimes have binucleated cells, so cells with 2 nuclei in them. So if we look closely, we can see a few cells that look like maybe they have 2 nuclei in them. Nuclei is the plural of nucleus, of course. So that's one way we can tell. It looks like it's several layers thick. It's very rare to have stratified cuboidal that's more than 2 cells thick. So that's a clue. And then when it looks like maybe it's several layers, much more than 2, and maybe a mix of cuboidal and columnar, that's definitely transitional. So look for double nucleus, look at the shape of the cells, count the number of layers. And your teacher might be really nice and give you a hint. What is this tissue that's found in the lining of the urinary bladder? And you would say like, oh yeah, I remember that's that weird transitional stuff. So that's transitional epithelium. All right, and in the next section, we will look at different glands and how they secrete their products.